Ladies and gentlemen, welcome and thank you for joining today's conference, African Swine Fever Clinical Presentation and Diagnosis. Please note that all lines will be muted until the Q&A portion of the call. We will provide you with instructions on how to ask a question at that time. You are welcome to submit written questions during the presentation, and these will be addressed during Q&A. To submit a written question, please use the chat panel on the right-hand side of your screen and choose All Panelists from the Send to drop-down menu. If you require technical assistance, please send a note to the event producer. With that, I'll turn the call over to Liz Fernandez. Liz, please go ahead.
Good morning, everyone. I am Liz Fernandez with the Professional Development Services Branch, and I'd also like to welcome you to our webinar today. Dr. Gregory Mayr is our speaker. Dr. Mayr is the Diagnostic Services Section Senior Diagnostic Advisor here at the Plum Island Foreign Animal Disease Diagnostic Laboratory. Greg is responsible for supervising and participating in the everyday functions of the ASF Diagnostic Services Section and overseeing coordination in its staff efforts. Dr. Mayr earned his PhD in molecular microbiology at the State University of New York at Stony Brook, and prior to joining ASF, Dr. Mayr held the positions of Associate Scientist and Research Associate Microbiologist with USDA Agricultural Research Services at PIAC. I will now turn the webinar over to Dr. Mayr.
Good morning. Hopefully today, as we walk through this presentation, you'll get a feeling for just how bad African swine fever could be if we ever saw it in our domestic swine population here in the United States. And if you have any questions, please, at any point, go ahead and send a note so that they can get that in the queue, and I'll try and answer any questions you might have. So we're going to talk today about clinical presentation. What does African swine fever look like? And then we'll talk a little bit about what kind of diagnostic tools do we have and how can we go about seeing whether or not this disease appears in the United States. So things you should already know about African swine fever. African swine fever is a swine-specific disease. So unlike foot and mouth, which will affect all ungulates, ASF is only a swine disease. However, it will affect all types of swine. So you will have it in feral pigs as well as domestic pigs. ASF causes rapid high morbidity and mortality in naive domestic swine populations. Most of the time when you see an outbreak, you are conscious of the fact that something is going on because there's a large die-off of pigs very rapidly. Now, there are some indications that as the disease gets itself set in the environment. So when you have an outbreak in a country, initially a lot of pigs will be sick, but then as the disease progresses through that country, you'll start to see it develops into more of a moderately virulent stage where the animals get sick and they live longer or they don't get sick as easily. And there's a lot of stuff going on with ASF, so it's a little bit difficult to discuss how that happens or why, but the initial inception of this disease into a population is likely to have high mortality. Feral swine populations may show fewer clinical signs and decreased or delayed disease severity. In general, feral swine are a little bit healthier in terms of their ability to resist disease, and it has to do with the constant disease insults that they see on a daily basis. Those animals also have developed evolutionarily to be a little less susceptible to the virus to the point where it will attack them and do damage to them, but they may survive long enough to spread it. African swine fever virus replicates and spreads in soft tick vectors too, and it can stay dormant in those tick populations for up to 8 years. So we have to worry about the fact that we have at least 2 soft tick populations in the United States that have the ability to support that virus if it made its way here. Other things you should already know. African swine fever is spread to China, and it's causing huge concerns for their pork production systems and for their trading partners. And they are a major consumer of pork throughout the world. So really the fact that their population is suffering from this disease now could have large ramifications for pork supply. African swine fever has been spreading through Eastern Europe as well, and it's been carried by the feral swine population. In 2010, there was a paper written by a biologist, an epidemiologist, who thought that feral swine populations were something that people weren't watching in the Ukraine, and that the pigs in the Ukraine had active interactions with all the feral swine all the way through to Poland. So they suggested that people should be wary of the fact that the disease was likely to spread from the Ukraine to Poland via the feral swine population. And that's exactly what's been seen. The most recent outbreak was in Belgium, and again, it was seen in these feral swine populations. So a little bit about the host range. Originally in Africa, the host range was the warthogs, the bush pigs, and the giant forest pigs. And these animals don't really show clinical disease. Over time, they've evolved to the point where the virus will grow in them. They'll be able to replicate the virus and pass it along to other pigs or to tick vectors, but they themselves don't show disease. However, the hosts that demonstrate disease include both domestic pigs and the European wild-born American feral swine. So we have to be conscious of the fact that even though the European wild-born and the American feral swine are probably less likely to show a severe clinical presentation, they are going to show clinical presentation. The domestic pigs are very susceptible. And again, keep in mind that this virus grows very well in a tick vector. So not only are they carrying it, but they're replicating it, passing it along to their young. They pass it through all the stages of their life. They pass it to the eggs when they lay the eggs. And it's also a sexually transmitted disease in ticks. So transmission, how does it happen? Well, contact between sick and healthy animals. That's a really big part of this. That's especially important when you start to talk about the feral swine population. Feral swine have a unique ability to get around obstacles placed in their path. So they're constantly damaging farms through fences, around fences, over fences. It has been a real problem in the United States to deal with these feral swine populations. If they get in contact with domestic pigs, they can spread the disease. Indirect transmission, garbage feeding, well, that's our biggest worry. A lot of times what has been seen is outbreaks will occur because people will be feeding things like sausage products that had gone bad and they no longer felt like they could eat them as humans, but it's okay to feed that to your pig. And if you're not cooking that properly, you're not killing that disease in that garbage, then there's a high chance that you're passing on whatever's in there. ASF is infectious for up to 3 to 6 months in that uncooked pork. Biological vectors such as the soft ticks are a problem. Again, if it made it into a soft tick population, we had a difficult time of getting rid of it. And fomites, including vehicles, implements, and clothes. So again, much like foot and mouth, when you have veterinarians going out to the farms to look at animals and see whether or not something is going on, you have to be very, very conscious of PPE and following the appropriate procedures to make sure you're not taking anything off that farm to any place else you might be going. And as I mentioned a minute ago, within the tick vector, it persists throughout all the stages of the tick's life, and it's transferred to the eggs and sexually to other ticks. So this slide I put in here just to show you a little bit of what's been going on in Europe. What I want you to be conscious of is the fact that you have two sets of columns here, the notification of wild boar cases in 2017 or 2018 and domestic pigs in 2017 and 2018. And what you can see is that the outbreaks in domestic pigs happen when you have a large population of wild pigs that are sick. Those wild pigs are what's moving this disease around from country to country. The only one that you're not seeing that is Romania, and we're not entirely sure that they were looking at feral swine when they knew about the two domestic outbreaks. We're not sure if they're tracking dead feral swine or not in Romania. The two outbreaks that they had in domestic pigs, though, resulted in more carcasses than they knew what to do with. So really, this is a very nasty disease and rapidly progresses through a domestic population. So clinical presentation. The clinical presentation that I'm going to describe to you is going to sound exactly like I'm talking about classical swine fever. You get the fever and the anorexia, the listlessness. If the pigs are pregnant, you get abortions. You get high fever, so you have huddling and piling of the swine because they're trying to stay warm. They develop respiratory issues, high respiratory rate, coughing, shallow breathing after a while. They have vomiting, constipation, then followed by bloody diarrhea. And the bloody diarrhea is really a very obvious clinical sign that you could be looking at African swine fever in those pigs. They get ocular and nasal discharge just like you would see for classical swine fever. Other clinical presentations, you see a reddening of the skin leading to cyanosis. So the extremities, the legs, the ears, the snout, you start to see that reddening of the skin followed almost immediately by it turning purple to the point where you can tell that the skin is not getting circulation properly and that there's hemorrhaging going on. You get incoordination and paddling 24 to 48 hours before the animals die. So it progresses into the brain and you start to see these neurological signs. And death usually happens in domestic pigs in 6 to 13 days for most of the strains that are out there, although there are some strains where it can take as long as 20 days. Rare survivors can be virus carriers for life. So again, much like classical swine fever, if the pig lives through this, it's now a virus factory that it goes on progressively spreading that to other pigs. So you can find various charts like this one that I've presented here in any number of publications. And they'll just take you through what you would see in the different types of clinical presentations you might see in pigs. The para-acute and the acute talk about that very quick disease where the animals get sick very quickly, go through a rapid progression, and then die. The moderate is what you tend to see after an outbreak has been going on for a while, and the pigs and the virus sort of adapt to each other, and you get this population of animals that are living progressively longer through the disease. And that chronic ASF is exactly that. Those animals have learned to tolerate the virus, whether it's because they got it at a lower dose initially and were able to form a partial immune response, whether you have some sort of herd immunity going on, or whether the virus itself is now using different genes. ASF is a very complex virus. It's a very large DNA virus, and it's got whole gene families. I know it has at least 7 different genes that it uses to avoid the pig's immune system. But it's not necessarily producing all of those proteins all the time. So various variations in that protein production of the ASF virus in those animals might be what's allowing this chronic form to develop. So what does it look like? So these are pigs that were infected with a very virulent strain of the ASF virus. You can see the hemorrhaging in the extremities, the legs of the pig all the way on the left, and its ear is bright purple. And you can see the bloody diarrhea on the floor there too. At a slightly earlier stage in the disease progression, you'll see that the pigs are up and moving, but they already have developed that bloody diarrhea. And lastly, when they're at the point where they're no longer functional and all they really want to do is just lie there and sleep, very rapidly death happens. So I've got a little video that I want to share with you, and this is a progression of African swine fever in pigs. Now the Lisbon 60 isolate is a pretty virulent strain, and these are animals 4 days post-infection. And you can see that they're pretty happy. They're walking around, they're eating. There's one pig sitting down there, but it's really hard to say, you know, if anything is clinically going on since it's eating. By day 5, you start to see subtle variation in that behavior. And now the pigs in general are moving about less dramatically. They're a little more sedated. The animals are now again sitting or lying down, knuckling, because they don't want to move around. They're having difficulty. By the time you get to 7 days, which is only 2 days after what you just saw, the disease has progressed severely. And now most of the pigs are lying down and are starting to show the hemorrhaging on the feet, on the snout, and up in the ears. You can see the sort of purple discoloration of the snout on that pig. And even the pigs that are looking a little bit healthier, and there's a good example of the ear hemorrhaging. Even the ones that are a little bit healthier looking, they're not moving about, they're not as energetic. Overall, they just want to lie there and try and stay warm by piling. And then a day later, it's even more dramatic. So now you can see the purple discoloration as we've gone from that hemorrhaging to the severe petechiation in the ears and in the extremities. Bloody diarrhea is going on still. And even if they get up, they have difficulties with coordination and they rapidly go back down. Now, this particular virus kills them at day 8. So these animals did not suffer long, but it was very dramatic. So when we look at those things, the things you have to keep in mind are that the presentation of ASF is very similar to a whole bunch of other things that happen in pigs. The clinical presentation for ASF is very similar to some of the more virulent strains of classical swine fever. Clinical signs are also very similar to other viral and bacterial infections in swine. So there are various bacterial infections that will cause a lot of those same similar clinical presentations. Coumarin-based poisons cause similar clinical presentation, and certain fungal toxins can do that as well. So there's this difficulty between is this ASF or not, and to definitively identify ASF, you need to be sending samples to a laboratory for testing. That's really the only way to know. So when should you be thinking about ASF? Well, you need to be looking at that pig population from the perspective of what am I seeing today that was different from what I saw yesterday. And in the video, you get a good example of how quickly that progresses. So one of the things that we do with classical swine fever when we're teaching our vet schools is we try to get them used to thinking about scoring the clinical condition of the animal population when they're looking at it. And I have score charts on the next few pages that were developed originally for classical swine fever, but they give you a feeling for how you might want to think about scoring pigs when you're looking at them to decide whether or not something is going on. And this first chart talks about liveliness and body tension and body shape. What does the pig look like? Is it healthy? Is it happy? Is it moving around? Is it having difficulty breathing? Does it look like it's emaciated, like it hasn't been eating? Are there central nervous signs? Is it having trouble with its coordination? Is it having difficulty walking? Does it have respiratory problems? Is it having, distinct chest movement? It's having difficulty breathing? Are there those hemorrhages going on in the skin? Do you see it in the ears and the nose and the legs and the tail that you have that, hemorrhaging going on? Are the eyes looking infected? Do they have that conjunctivitis? And I realize conjunctivitis is something that pigs get for any number of reasons. But if you're seeing that in conjunction with some of these other clinical presentations, you might be thinking, okay, we should call somebody and get a foreign animal disease investigation going on. Appetite, again, if they don't want to eat, if they're so uninterested in eating that they're just lying there while you're putting food down, that's a bad sign. And again, the defecation. In African swine fever, that bloody diarrhea is a real tell that that could be what's going on in those pigs. So things you might know about ASF. Well, it can be found in whole blood. That's a really good sample for African swine fever. EDTA blood can be used for PCR testing, and heparin blood can be used for virus isolation. Tonsils, spleen, lymph nodes, those are good internal organs. They have a lot of blood flow. They have interaction with the blood system. So the virus will pool in those organs. It actually grows in some of the macrophages that are part of the tonsil. And so you will get virus if you're looking in those organs. Some of the more internal organs can also have traces of African swine fever. I know that in a lot of the feral swine studies being done in Europe, they're using bone marrow because the truth is when you find a dead feral pig, its buddies have already eaten all the good parts, so the only thing left is basically the skeleton. So having no other sample to use, they'll look at bone marrow. Is it the best sample? No. Is it a sample that they have available? Yes. Survivors are rare in domestic swine, at least at the beginning of an outbreak. So you want to be sure that if there are survivors, you're looking at them for antibodies. And so any survivors that are present, test the sera for antibodies. So what do the tissues look like? Well, tonsil tissue looks better than it would for classical swine fever. The necrosis in classical swine fever is much more dramatic than what you would see in an ASF infected animal's tonsil. So overall, the tonsil looks a bit healthier. When you look at the spleen, however, you can see that the ASF-infected spleens are much, much bigger than the normal spleens put next to them. It also has this friability. You can bend the spleen over and it sort of snaps. So it's very, the damage done in the spleen is significant. ASF can be found in the lymph nodes. You'll see the hemorrhaging in the lymph nodes and the swelling. Most of the time when people are looking at a dead animal and they open it up and it's African swine fever, one of the first things they'll see besides the very swollen spleen is the fact that all of the lymph nodes are enlarged. So what do we have as our toolkit to use here at Saddle and in the Nolan labs to look at whether or not this disease is happening? So here at Saddle we have virus detection through both PCR and virus isolation. Virus isolation can take up to 21 days, though. It's not a rapid process. The cells that you're using are porcine monocytes or macrophages. So you get those from a bleeder pig, a donor pig, and then you infect them and you look for cell death and the ability to hemagglutinate. ASF is the unique virus in that it has that ability to cause hemagglutination. PCR is a really quick and good test. It takes about 2 hours. So on whole blood or tonsil or spleen, Saddle is going to look at those to see whether or not the PCR is positive for African swine fever. The protocols that went out to the Nolan labs a couple of years ago to look at a possible surveillance were with whole blood, and that's why whole blood has been the sample that the Nolans have been previously approved to use for testing for African swine fever in pigs that are sick. Immunodetection. We have the ability to do cryosectioning of the spleen or the tonsil or lymph nodes and use monoclonal antibodies to look for the presence of the virus. And that immunostaining takes about 4 hours. It's a very nice tool to use if you have a decent tissue sample. Serology. Well, the ELISA kit is a purchased kit. That's the downside. It's a very good kit. It gives you nice answers, but they come from Spain, and the production facility in Spain is not designed to supply kits to the whole world, although that's what they've been tasked to do for the past 10 years. So you can get kits, you can't get them quickly, and you can't get a lot of them, but you can get kits. If you get something that cross-reacts and you say, why do I have this borderline questionable answer on my ELISA? Unlike classical swine fever where we have an immunoperoxidase test followed by a virus neutralization test, all we have is an immunoperoxidase test, and we've only been using that for 2 or 3 years here at FAO. We have an IFA test. It's an immunofluorescence assay test that we have been using before that, but the reagents for that are limited. So if we had a real outbreak, we would rapidly run out of those reagents. The immunoperoxidase test is currently undergoing validation here at FAO. As a nation, what are we doing to make sure that ASF isn't coming into this country? Well, I can tell you that up until recently, most years, we were only getting 3 to 4 cases a year where we were requested to test for classical swine fever or African swine fever. In 2017, it was only 3 cases. In 2018, we're up to 7. It's doubled. Most of those 7 cases have happened in the last month since they announced that China has African swine fever. African swine fever serological surveillance, there's currently no program. We're doing CSF surveillance, but not ASF surveillance. And ASF PCR surveillance, currently we have no ongoing program in the United States, but this is under consideration, and it's being decided way up the chain whether or not this is something that we want to get involved in and go down that road and decide to have an active surveillance program in the U.S. So points to remember. ASF is likely to have very obvious clinical presentation with high mortality, at least at the beginning of an outbreak, and abortions in pregnant sows. Recovered animals, including wild swine or domestic pigs in zoonotic areas, may become persistently infected virus carriers for life. Those pigs have the ability to spread the disease continuously. And misdiagnosis as something you would normally see, you know, if you're looking at pigs at a slaughter plant or you go out to a farm and you're looking at pigs and you see things like strep infections in the pigs, you can get a lot of those same clinical presentations. So if you wait and you say, oh, no, this is strep, this isn't ASF, and you don't send samples right away, there could be a delay in diagnosing this for what it truly is. If ASF becomes established in a soft tick vector, again, that's going to complicate how do we go about getting rid of this disease and proving freedom. And only laboratory diagnosis can definitively determine that ASF is causing the clinical signs that you're seeing in animals. So please, encourage people who are out in the field that if they see something going on in pigs and they're not comfortable with how it looks, please send samples for diagnosis. I want to take a moment and thank Dr. Peter Fernandez. Peter worked for the USDA for many, many years, but he's happily retired now. But a lot of these slides came out of presentations that he's been using for the past 10 years to help teach people about ASF in foreign animal disease diagnostician schools. And with that, I'll take any questions you might have.
Ladies and gentlemen, as we move to Q&A, please press #2 on your telephone keypad to enter the question queue. You will hear a notification when your line is unmuted. At that time, please then state your name and question. Once again, pressing #2 will indicate that you wish to ask a question. To submit a written question, please use the chat panel on the bottom right-hand side of your screen and choose All Panelists from the Send To drop-down menu. Thank you.
So we do have a couple written questions. The first one is, are peccaries affected by ASF?
That's a really good question. I don't technically think they're a pig, but I have to look into that. I know that they do not get CSF, but I don't know about ASF. I'll have to look into that and get back to you.
And the next written question is, in the case of chronic ASF, would you expect to see erythema/cyanosis in the absence of other signs?
I have to tell you that most of the work that's been done with low virulent strains has been by deliberately mutating the virus so that it causes less damage in the pigs and you can go ahead and work with it. It's very difficult to get a field strain that's a low virulent strain and introduce it in truly naive pigs and not see a lot of damage. That being said, you'll still see that hemorrhaging and the erythema, but not as dramatic, and the hemorrhaging will happen in the internal organs, so you'll still see damage in the spleen. So if you had sudden death, take a look at that spleen. Whether or not you see the hemorrhaging as badly as you do in the external, on the legs or in the ear or anything like that, it might look a little pink, but it might not be as clinically obvious before the death of the animal.
So the next written question is, there is some thought that feed may be a pathway for introducing the ASF virus. What is your thinking on this?
Well, okay, so if you're talking about commercial products versus garbage feeding, if you are feeding garbage to a pig, it should be properly cooked. That's your best way to go about avoiding introduction of any type of virus, whether it's PEDV or ASF, to those pigs. If you are talking about products that are being purchased from countries that have ASF, we have no way of knowing what the density of virus in those products might be, no way of testing for it. It is not, that's not an easy question. I mean, the truly stupid answer, if you'll pardon me, is that if you continue to feed that kind of stuff to your pigs and they get sick, that's how we're going to know that that material was infected. Because truthfully, the amount of volume of the material you'd have to test in order to have a representative amount tested to say whether or not it was free of the disease is almost impossible.
So the next question is, how are you disposing of the ASF animals?
Well, that's another great question. Over in countries in Europe, they're burying them and burning them. The virus is pretty stable, so burning them tends to allow you to get rid of the virus relatively quickly. I know that the virus is stable over a large pH range, but if you bury them with enough lye, you cause the pH to go high enough that you get rid of the virus. Is it environmentally friendly? No. How would we deal with it here? That's a great question, and I don't have an answer for you.
So we have a compliment first, great presentation. Can you discuss the current U.S. classical swine fever surveillance, and can this easily be extended to include testing for both ASF and CSF?
So the current surveillance that goes on in the United States for classical swine fever involves both serology. Here at FATL we use commercially available kits that are made internationally, which is why we use them here as opposed to in the Nolan labs, because up until very recently they contained select agents, so they had to be handled in a facility like FATL. So we do the ELISA work for looking for classical swine fever, and we run those kits, and then we do the IP and the virus neutralization test if we get something that's cross-reactive. If you get a high enough titer in a pig against BVD, you'll see classical swine fever cross-reaction on that ELISA. For ASF, it's a little more complicated. As I said, currently we don't have a serology surveillance program in the United States, and are we likely to go there? I couldn't begin to tell you. Part of the problem is ELISAs are not a very, unlike PCR, which has high sensitivity and specificity, ELISA kits tend to have a much lower specificity. So you'll get a reaction to something that looks like it's something that generates an antibody similar to what you would see for classical swine fever or African swine fever can cross-react on that test. Like I just mentioned, BVD for CSF. So we don't know what domestic conditions are out there in the environment, whether they're bacterial or viral in nature, that could cause a cross-reactive answer on the ELISA kit. We just don't have the data for it. So it's very difficult to start to go down that pathway and say, okay, let's start a surveillance program for looking at serology, because it becomes very difficult very quickly to make sure that what you're looking at is a domestic cross-reactivity and not a real answer on an ELISA. PCR is a little easier. So PCR and a surveillance program, that's being considered, and that's why that's being considered, because it's very sensitive and it's very specific. So if you see something and it's positive, it's real. But again, that has trade implications. So it's a very complex, from a science perspective, it's very easy. But from a trade perspective, it's very difficult to say, should we be looking for something that we know currently doesn't exist in the United States, because, oh my God, what if we found something? That's from a trade perspective, that's a very difficult question.
We have another question. It's the status of ongoing research on vaccines for ASF, and why has it been so difficult to develop a commercial vaccine for ASF?
Unlike other viruses where you can use a killed vaccine, African swine fever has a whole, like I said, it has a whole family of proteins that the virus produces when it infects a pig that shut down various parts of the immune system. So you don't tend to see a real good immune response. Plus, there's very little data out there between the relationship between antibody response versus cellular response in preventing disease in pigs with African swine fever. The killed vaccines that they've tried have not worked. The ones that they're working on now are genetically attenuated, so they've gone in and they've changed some of the genes specifically to allow that virus to grow in the pig but not to cause any damage. So there are, ARS is currently working on vaccine candidates, and Dr. Manuel Borca gave a nice presentation, and at some point Liz can hook you up with that presentation so you can take a look at it. But they're doing really good work looking at these vaccine candidates and seeing whether or not this is something that we might be able to move towards commercialization of and get a product out there. But throughout Europe, throughout Africa, none of the things that they've tried as far as vaccines have worked continuously in a population.
The next question is, what NALM labs are able to offer ASF diagnostics at this time?
There are a handful of labs that have been proficiency tested and cleared for African swine fever PCR in the event that there's a sick animal. So we're not talking about surveillance, we're talking about if there's a case where the animal is sick. I think there are 10 current ASF approved NALM labs, but Christy Liacino could get you a list of which labs have that ability and which labs don't.
Okay. We have another question about active surveillance program. If it is decided to do an active surveillance program, what might that look like?
Okay. Well, that's a hypothetical question, so I'll give you a hypothetical answer. There are any number of ways to go. If you're looking at surveillance, in theory, what you're looking at is a population of animals that are healthy. So if you're looking at healthy animals, what sample types do you want to use? You could use blood. That's what was previously tried to show that healthy animals in the United States show no signs of African swine fever. And that works pretty good. In animals that are if you're looking at targeted surveillance, so you're looking at animals that are poor doers, they're not doing well, they have bad body posture, they have emaciated physique, they might have some hemorrhaging or conjunctivitis. Currently, those pigs are being sampled at slaughter. Their tonsils are being used for CSF surveillance. So they're currently looking into and debating whether or not we want to go down that road with African swine fever as well. Any animal that is sick and you think it might be a foreign animal disease, though, that should be an investigation. So we're not talking about surveillance. We're talking about there should be more submissions for an investigation.
Next question is, would composting infected carcasses kill the virus if the temperature reaches 131 degrees Fahrenheit?
I don't know the answer to that. I think it has to be hotter than that, but I couldn't begin to tell you how much hotter than that. If I remember correctly, the virus is very stable at temperatures up into the 150 to 160 range, but I would have to look into that and get back to you. I don't know that composting would work unless you're hitting the virus with multiple things. Let me take a step back. I should point out to you that the virus, if you're talking about virus in a test tube, it is more susceptible to being inactivated than if you're talking about virus in a bodily fluid or a tissue. In things like blood or tissue, the virus gains stability because all those proteins in that blood or in that tissue stabilize the virus against things like pH change, against things like temperature. So in a dead animal, that virus has a very long shelf life. So I can't even begin to answer the question of how long would it take to inactivate if you were that with the carcasses.
Okay, so we had another question about composting, but I think that actually answered that. At one time, I think ASF was present in Hispaniola, particularly Haiti. Has it been eradicated?
It was eradicated, I want to say, in the 80s. ASF is no longer in Haiti. That being said, nobody's looking anymore, right? Haiti has always been a problem from the perspective of illegal movement of people and product through the Caribbean. So is it one of those places where if we were to develop surveillance, we'd like to partner with them and look, or with them in the Dominican Republic? Probably. That being said, right now, ASF is not in that country.
Okay, I think we have a written question, so let's go ahead and take the written question next.
You said written.
I'm sorry. We have a verbal question. Thank you.
Thank you. Caller, please go ahead.
Yes, can you hear me?
Yes, I can.
Okay. You may have answered this, but my question has to do with out in the environment. Let's say a feral swine was, a hunter was to kill a feral swine and field dress this animal. The carcass is now gone, but we got offal and we have animals spreading this offal around. How long would that be found in the environment in a situation like that?
So in the parts of the carcass you're not taking home to cook, any meat left behind, any blood left behind is a source of contaminant that other feral pigs can come along and lick or eat and get infected. And that's stable in that environment, that along the same lines as the uncooked pork. You could see that in the environment for 3 to 6 months, depending upon the condition of the area, whether or not you're talking about the middle of winter, where it's stabilized because of the temperature, or whether you're talking about, Texas in the summer, where it's 110 degrees out there. The environment will play a role. This is an enveloped virus, so it's less stable when it dries out. But that being said, the proteins in that tissue, in that blood, can stabilize it even under those conditions.
Okay, do we have any additional verbal questions in the queue?
There are no additional questions in the verbal queue.
Okay, I have another written question. To take the mitigation discussion further beyond vaccines, is there any effort to harness the natural resistance of some wild suids looking forward to gene editing production swine to express the same resistance?
That's a really complicated question on so many levels. I know that there has been efforts recently to look at PRRS-resistant pigs that were genetically modified to get them to that state. And the FDA is now dealing with those because that becomes a genetically modified organism. So you have a whole series of rules and regulations you now have to follow because that's what that is. Plus, for whatever reason, even though we've been breeding animals for hundreds of thousands of years and domesticating them and making them what we want them to be by breeding them and getting them that way, if you go in and directly genetically modify something, for some reason the customer no longer wants to buy that product. So there would be a significant amount of GMO backlash on if you did that to the pig population. So I can't even begin to answer what that would do to the ability to sell pork in the United States. It's a great idea. As a scientist, I'm behind you, but there are all these other things that we have no control over.
Okay, our next question is, what temperature will inactivate the virus in cooked product?
Off the top of my head, I don't know. I believe we cook that product at like 160 or 165 degrees for 20 minutes, and that should be sufficient to kill the ASF. If you're following your state's regulations on garbage feeding, you're doing the job right.
What disinfectant products are recommended, labeled, approved for ASF?
Ooh, that's another good question. I think it depends where you look. From our perspective, things like Virkon or 10% bleach are good, but you have to be aware of the fact that if you're talking about cleaning up blood off a hard surface, that's great, but if you're talking about there's organs and blood on the ground, it becomes a different matter. Now you have to make sure that your environment is saturated, right? So it's not a straightforward question. You can disinfect. It's just, it isn't easy. And if you're talking about in a commercial production facility where they use, fluorine that can be decontaminated, you're talking about a much easier decontamination process.
The next question is, will testing be performed only if a FAD is suspected, or will there be surveillance testing approved for high-risk premises such as garbage feeding operations in Puerto Rico?
At this time, only FAD investigations are allowed to be tested. We're still waiting for word on how it's going to be, how a surveillance program is going to be sent out to the states and to the Nolan labs. So, again, that question is still up in the air. I wish I had a more definitive answer for you, but at this time, it has to be a farm animal disease investigation.
The next question is, in order to increase FADI submissions, there could be a resulting increase in quarantines on parts or all of large commercial swine facilities pending results on the FADI. What sample results are needed to confidently release a quarantine? Would we wait on VI?
Wow. I can't decide if it's fortunate or unfortunate, but I don't get paid to make policy decisions. From a science perspective, much like we're doing for, foot and mouth and classical swine fever, I think a lot of quarantine things very rapidly you would see a progression towards a PCR negative answer gets you release of the carcass. But I can't guarantee that. This is not my decision to make. Virus isolation with ASF is a very prolonged process, and that's because, especially if you have if the sample has a low virus load, it takes a long time for that infection to spread along the monocyte layer and develop itself to the point where you start to see cell death and hemagglutination when you have the red blood cells. So it could take, like I said, up to 3 weeks. I know when they're looking at, are you sure it's not there, it's 2 to 3 weeks of virus isolation to feel comfortable with the fact that there's nothing there. So in a case like this where you're talking about, are we sure there's nothing in this animal, I don't think they'd be waiting for virus isolation.
The next question is, would there be any risk or preventative measures needed for international visitors from ASF endemic countries visiting U.S. swine farms?
Well, the first step would be to make sure that they're not bringing anything with them. I hate to harp on it, but that garbage feeding is a huge problem. One of the recent messages that came out about the introduction into China had to do with sausage coming from Mongolia or Russia. So, and I don't remember which, and I apologize. But those products, those meat products, we have to ensure that they're not coming into this country, and I think our TSA people are doing a great job of helping prevent that. I've recently seen some images of things being confiscated in airports, and they're doing a heck of a job. That being said, I think that if you have people and they're coming from an area that they worked on a farm and they're coming over here to visit their friend who has a farm, we should consider telling them that they can't have been on their farm for the same sort of period of time that we require people who are leaving a place like Saddle who work on these diseases. So, a 5-day quarantine I think is something that it's really good advice. Making sure that you're not bringing again, because of the condition of this material in a sample that has bodily fluids on it, you want to make sure that their shoes are clean. So would it help to have disinfecting pools in airports where you walk through them? I know that there are countries that do that. That'll slow everything down. So everything's a trade-off. Again, these are not easy questions. From a science point of view, yeah, I'd have you walk through a Virkon bath and make sure that you didn't go out to the farm for 5 days, and I suggest that you have all your clothes washed before you go there. From a practical point of view, I don't know what regulations I can impose on people.
We have another question. It says, is anyone analyzing ticks in the U.S. for potential presence/arrival of ASF virus or other foreign animal diseases?
No. So ticks carry so little bodily fluid, if you will. You would have, it's kind of like doing surveillance in mosquitoes for West Nile. You have to collect a lot of mosquitoes before you can get enough material to do a PCR and say, yep, it's here. So I currently, when we don't have the disease in this country, nobody's looking in the ticks. Will they when we start getting disease, if we get disease in this country? Yes, it would not surprise me if people were starting to look at some sort of trap for ticks to determine whether or not that population had been infected.
How about survival in manure, the ASF?
Well, if it's bloody diarrhea, it's in the blood. So it's going to survive for a prolonged period of time. You're talking a couple of weeks.
How long would it live in a lagoon from a positive premise?
Again, the lagoon is an interesting question because you've got all sorts of things going on in that lagoon, including heating. And as the material breaks down, does it become less stable? Sure. How long does that process take? I have not seen any papers documenting a study that was done to look at that question.
What is Europe doing to control the environmental tick population that may be spreading ASF?
I don't know. I know that they have a problem with their feral swine. I don't know if they have a soft tick population, so I couldn't answer that.
This is a follow-up question. As a scientist, are you comfortable with the PCR negative result alone?
No. So, okay. PCR is a great tool, but it's only one tool in your toolkit. And PCR in a vacuum, it's a hard thing to say how valuable it is. If you're asking me if I had a spleen from an animal that looked like it has ASF and I did a PCR on that spleen and it came up negative, would I believe that? Yes. Yes, I would. But if you're asking me if I did a PCR off a swab and the pig looked healthy, does that tell me anything? No. No, it doesn't. So the circumstances surrounding that PCR really are important. It has to be in the proper context. It also helps to have observational surveillance. So if you're going to a farm and you're seeing dead pigs and the farmer tells you that all his pigs were healthy 5 days ago and now a large percentage of his population is dead, that's significant. That's a clinical presentation in an environment where he's seeing something he hasn't seen before. So it's a rapid onset, rapid progression. If you have somebody who says that they've got one dead pig, but they've been losing about one every 2 weeks for the past 8 months, is that likely to be ASF? Well, probably not. So now, if I get a negative PCR off that one, do I believe it? Yeah, I probably will, because the observational surveillance that goes with that diagnosis of negativity go together. So again, I think that we have to really use all of the tools at our disposal. So in large production facilities where I know that they have veterinarians who are assigned to oversee the health of those herds, that observational surveillance is really a beautiful part of this. If we start doing surveillance, and you can tell me that those pigs all look healthy for the past month, that goes a long way, at least from a science perspective.
Here's another question. Are there standard recommendations to mitigate the risk represented by students and grad students from countries with ASF outbreaks attending and working in our land-grant universities, vet schools, and ag research stations?
I would assume that they have the same, you can't go near any of our animals until you've been off your country's farms for at least 5 days.
Yeah.
If your university is not using those, you should start that process, whether it's FMD or CSF or ASF. You can't have them going from a dirty area to a clean area without some sort of quarantine. And I'm not talking about locking them in a closet. I'm just talking about don't let them go on the farm for 5 days.
So this is a comment. This person recently returned from Finland and Iceland through Canada. We were not asked anything about food when entering the USA. This seems like a large hole in keeping food products from entering the USA.
You should have had, while you were sitting on the plane coming back into the country, they should have handed you a declaration form. And one of the boxes on that form says, have you been on a farm or did you go to an agricultural establishment? Are you bringing any food? You probably are so used to checking the boxes that you check no without even realizing, but you should have gotten one of the, every time I go out of the country, I get one of those forms. And as a USDA person, I then stand in line at customs and they say to me, you work for the government, why didn't you tell us? But yeah, no, you should have gotten one of those questionnaires.
And this question, did you say disease is passed via mosquitoes?
No.
West Nile.
No, West Nile, not African swine fever. Although nobody's ever looked at that. But it is, it grows in the ticks. I think that's why the ticks are such a really good vector for this disease, because not only are they carrying tiny amounts of blood, but they're also growing the virus in their salivary glands. I have beautiful electron micrographs of hundreds and hundreds of virus particles growing in an individual cell in the salivary gland of a tick. It's just beautiful. So, yeah, they not only carry it, they amplify it. So, you know, I don't know that people have looked in mosquitoes or things like that, but that is, at this time, that is not one of our concerns.
Okay. Do we have any other verbal questions in the queue?
There are no additional questions in the queue.
And as of right now, we have no questions in the written chat boxes. We went through them all, and thank you for all your questions. I don't think I've ever read so many questions on a webinar. So there's one comment. The question did ask what you said, but when they came through Canada, but then asked if I was going to visit any farms in Canada. Allows one to answer no, then at U.S. border, no question.
Interesting.
That is interesting. I don't know.
Well, I guess because Canada's clean, so we assume if you're coming from Canada, you're clean. But that strikes me as very odd.
Yeah. But I flew from Ottawa to USA, and I had to stop in Baltimore, and I thought I was going to have to pick up my luggage and go through immigration and everything, and I didn't. I just was right in the terminal.
Interesting.
So, I don't know.
Well, thank you for that comment. We'll have to look into that.
Yeah, we will have to look into that. So, well, if we have no other questions, I want to thank Dr. Mayr for his presentation and thank everybody for joining us today. We had a record number of people on this webinar, so that's very exciting. We do have another webinar going to be held on October 18th, which is next Thursday at 11 a.m. Eastern Standard Time, and that topic is the state's perspective of operationalizing the Secure Milk Supply Plan. So with that, I will say have a great afternoon, and hopefully we'll have you back on next Thursday.
Thank you all for listening.
Thank you for joining today's presentation. The event has now concluded, and you may disconnect.